The International Barcode of Life Consortium is developing DNA-based identification systems to fast-track biodiversity assessments. Launched in 2010, it now includes research organizations in 40 nations with a shared mission to activate a global biomonitoring system. This goal is motivated by the massive declines in biodiversity over the past 50 years. Biotic change can't be tracked with a single technology. Remote sensing is a powerful asset, but it must be reinforced with ground-based programs because many essential biodiversity variables can't be tracked from the sky. For example, only 5 of 12 essential biodiversity variables for oceanic ecosystems can be partially evaluated with satellites. The IBO Consortium is dedicated to realizing the potential of DNA-based identification systems to advance our capacity to track EBVs on land and in water. To achieve this goal, it's coordinating 3 large projects. Barcode 500K is complete, while BioScan is in progress, and the Planetary Biodiversity Mission will launch in late 2028. DNA-based identification systems come in 3 flavors, depending on the source of the DNA extract targeted for analysis. DNA barcoding involves analyzing extracts from single specimens, while metabarcoding analyzes DNA from bulk collections. Finally, studies on DNA extracted from air or water are termed eDNA. Whatever its source, analysis focuses on the sequence characterization of a tiny fraction of the genome. In the case of animals, most species can be discriminated by sequencing a 650 base pair segment of their mitochondrial cytochrome C oxidase 1 gene. That's a millionth of their genome. Once you have a sequence, its source can be ascertained by finding its match in a library of reference sequences obtained from identified specimens. DNA-based systems have gained tremendous power over the past decade because of remarkable advances in sequencing technologies. Long-read platforms, such as a sequel, can gather barcode records from 40,000 individual specimens per run, while short-read platforms, such as NovaSeq, can ascertain the species present in 500 bulk samples per run. Through their use, a specimen can now be barcoded for $1, while species occurrence data can be generated via metabarcoding or eDNA for $50 a sample. Because the latter samples can include 1,000 or more specimens, the cost per individual is low. Let me use a metaphor to emphasize the critical need for a DNA barcode reference library. Trains can't reach their destination without a track. In the same way, DNA metabarcoding and eDNA studies can't deliver a species identification without access to a barcode reference library. The IBO Consortium is striving to complete 3 tasks before mid-century. It aims to register every multicellular species. It seeks to document their interactions. Finally, it plans to activate a global biomonitoring program. Let me provide a status report. Launched in 2010, Barcode 500K had a single goal: to assemble a DNA barcode reference library for 500,000 species. It met this target by late 2015 and laid the foundation for further progress by developing lab protocols to process 1 million specimens a year. It also developed the BOLD informatics platform to ingest the resultant data and the barcode index number or bin system to automate species recognition in the animal kingdom. Let me quickly describe what's involved in barcoding. DNA is extracted from the whole specimen, or part of it, and the barcode region is then PCR amplified and sequenced. Once the sequence record is coupled with an image of the specimen and details of its collection location, the barcode record moves to BOLD. As each barcode record is obtained, it's assigned to an existing bin, or, if it has a novel sequence, it founds a new one. Bull now includes records like this one for more than 750,000 bins. Their effectiveness as a species proxy transforms our capacity to study groups without well-developed taxonomy. By coupling DNA barcode analysis with the bin system, it's now possible to gain a comprehensive species count for any species, any site. You simply collect specimens, sequence the barcode region, and assign them to bins. The barcode map from 500K seems impressive, but it's just the first step towards a global biomonitoring system. Barcode 500K laid track, but only for about 5% of multicellular species. Bioscan is building on this foundation. It's expanding the reference library by employing the bin system to delineate species. It's using long-read sequencers to reveal species interactions. And finally, it's employing metabarcoding and eDNA to examine species dynamics. There's an analytical target for each theme. Barcode 10 million specimens, examine the symbiomes of 1 million specimens, and analyze 100,000 samples. Because of time constraints, I'll only discuss themes 1 and 3. Theme 1 is expanding the DNA reference library through 2 approaches, skims and deep dives. Each skim involves the analysis of 10,000 specimens from a site, and this work's underway at 500 localities. A third have now been analyzed, and this work has revealed 213,000 species. Another 5 million specimens will be analyzed from the Arctic, from Europe, and from Costa Rica. The effort to gather DNA barcodes for every species in Costa Rica was recently approved by its cabinet as an important path towards the protection and development of its biodiversity. Theme 3 is exploring species dynamics by using metabarcoding, or eDNA, to analyze the species present in 100,000 samples from 2,000 sites, positioned in half the world's 867 ecoregions. If these samples contain an average of 1,000 specimens each, this theme will analyze 100 million specimens. Although many bioscan projects are underway, I'm only going to be able to describe 5. 2 involve the development of biodiversity baselines. 1 involves a resource development project, and the others involve sustainable agriculture. Biodiversity baseline studies target protected areas to ensure that long-term change can be evaluated. Canada has 17 ecoregions, and the smallest, the Mixed Woods Plain, was the first to be analyzed. South Africa has 19 national parks, and the largest, the Kruger, was examined first. Both studies focused on arthropods, the most diverse component of terrestrial life, and employed malaise traps to collect samples. These passive sampling devices are easily deployed, and each trap collects from 10,000 to 100,000 specimens per year with little effort. Typically, the catch is harvested weekly, a task that requires about 5 minutes. Work in the Mixed Woods Plains began with the deployment of a malaise trap in 45 provincial parks. The sequence analysis of 1 million specimens from these sites revealed 25,000 species and made it possible to quantify the extent of species overlap among localities, while also generating the baseline data needed to evaluate biotic change through time. Collection programs in the Kruger were led by park rangers who ran traps at 25 sites in both its ecoregions, the Low Veld and Mopani woodlands. Analysis of nearly 300,000 specimens revealed 20,000 species. These results showed that species richness was higher in the Low Veld than woodlands, and that insect communities corresponded to established ecoregion boundaries. Imagine a future where we have similar baseline data for every national park and for every ecoregion. The resource development study is examining the impacts of a geothermal facility on the shoulder of a volcano adjacent to the ACG, a UNESCO World Heritage Site. Costa Rica's National Electrical Commission, the Japan International Cooperation Agency, and staff from the ACG joined forces on this project, and we did the analysis. 9 malaise traps have now been deployed for nearly a decade along 3 transects at this site. The DNA barcode analysis of 1.5 million specimens revealed 36,000 insect species and demonstrated that the impacts of this facility on biodiversity are modest. Shifts in species abundance and richness are restricted to the clearing. 50 meters into the forest, there's no perceptible change. Although this project was implemented to evaluate the ecological impacts of this development, it's expanded the barcode reference library. As such, the results are not only relevant to Costa Rica, but to nations throughout the New World. McCain Foods has launched its Farms of the Future initiative in 2020 to improve the sustainability of potato production, the world's 3rd most important food crop. The first farms in Canada, but companion sites will soon be activated in Africa and South America. Studies at each location will examine the impacts of agricultural practices on biodiversity and their linkages to factors such as the organic content of soils. Dole is striving to improve the sustainability of pineapple production. This crop currently occupies 1% of Costa Rica's land surface and is rapidly expanding. Conventional cultivation employs heavy applications of biocides, but Dole's now developing organic farming methods. DNA barcoding is being used to compare the impacts of these 2 practices on insect biodiversity in the fields and adjacent forest corridors. Canada has provided $150 million to develop the Centre for Biodiversity Genomics as the world's first DNA barcode core facility. It occupies 50,000 square feet of purpose-built space in 2 buildings, one constructed in 2007 and the second in 2012. It has a staff of more than 100 researchers, technicians, and informaticians. Its sequencing unit barcodes more than 1 million specimens each year and metabarcodes thousands of bulk samples. Its informatics unit has developed 2 key platforms. BOLD supports the storage, curation, and analysis of DNA barcode records, while Embrave aids the analysis of data gathered by high-throughput sequencing platforms. The CBG provides analytical services to hundreds of academic, governmental, and private sector organizations each year. Let me summarize. The transition to DNA-based biomonitoring has several value propositions: increased scale, increased objectivity, and lowered costs. These benefits reflect the fact that collection methods are simple, and the DNA sequencers can process massive numbers of samples. Informatics platforms, supported by public funds, are available to seamlessly capture and analyze data. Taxonomic subjectivity is avoided by the bin system. Importantly, the results can initially be held private, but can then move into the public realm with a simple command, escaping the gray literature trap. Let me finish with a short video on Bioscan.
Our living planet, a library of life. Every species like a book, holding the information for humanity's greatest innovations. New medicines, technologies, and economic development. But we are burning those books before we've even read them. We want to know all life, every species. We are the International Barcode of Life Consortium, and we are illuminating biodiversity through DNA-based analysis. By using automation and rapidly evolving DNA sequencing technology, we are bringing this vision to life. Our new scientific program, BioScan, is shedding light on unknown species, tracking their distribution, abundance, and interactions. BioScan will bring us closer to building a complete library of life, forming the basis for an Earth observation system. Together, we must act. We are not just scientists. We are rangers in national parks collecting the data required to monitor ecosystems. We are Indigenous peoples protecting our lands and traditions for future generations. We are citizens supporting science and conservation in our country. This is BioScan, collecting, mapping, and revealing life's diversity. Our species acting to protect all species before we lose the possibility of even knowing them.